Which of the following is not a common feature of DNA binding proteins? Pause the video and try out this question for yourself. Now, option number A says beta sheets sandwich the DNA strand. What do we think? Well, actually, the vast majority of DNA binding proteins, they don't use beta sheets to sandwich the DNA. Instead, the most common structural motif for sequence-specific DNA binding is, you guessed it, right, the alpha helices, right? So it's not beta sheets, but rather alpha helices. And that's because the alpha helices, it fits the major groove pretty well. And it's specifically I'm referring to the major groove of B form DNA. So fits major groove of B form DNA. And that's because it fits it very well, right? And like common motifs like helix, turn helix, zinc finger, and leucine zipper all rely on alpha helices to read DNA sequence. So, well, option number A is correct, but we also want to know why B, C, D, and E is not correct. In other words, why are they common feature of DNA binding proteins? So option number B says binding to a specific sequence occurs in 2 steps, finding the DNA, then finding the correct sequence. And that is a common feature, right? Binding does occur in 2 steps. First, you have to be able to find the DNA, right? That's because the nucleus is crowded and the genome is massive, and the protein cannot just rely on random 3D floating to find its exact sequence. And instead, it first have to bind DNA somewhere, non-specifically, right? So that's step 1. So find the DNA by binding it to it non-specifically. And second, then it's going to perform a 1D search by sliding along the strand until it chemically recognizes its target sequence, and then that's step number 2. So that's how DNA binding proteins work in general, right? So this one wouldn't be correct. Option number C tend to have a lot of arginines and lysine. Well, arginines and lysines, they are positive charge, right? So positive amino acids. And we know that sugar phosphate backbones is heavily negatively charged because of the negatively charged phosphate. So, in other words, DNA is negatively charged. And negatively charged, that's going to pair well with the positive amino acids, right? So that is why the answer is not C. And then option number D, they slide along the DNA until they find the correct binding site. And that's exactly, that's analogous to option number B, right? So it also wouldn't be correct. Sliding along the DNA, so it first finds the DNA, it slides along it until it finds the correct binding site. In other words, it finds the correct sequence, right? Option number E, DNA binding proteins can recognize specific DNA sequences that are visible within the major groove. Well, the major group, we know it's quite wide, right? It's wide enough to accommodate protein domains. And so yes, they do recognize specific DNA sequences that are visible within the major group because the major groups have those exposed edges of nitrogenous base, which creates barcodes of hydrogen-bound donors and acceptors, right? And proteins are able to read those unique hydrogen-bound donors and acceptor from those nitrogenous bases and be able to find their specific sequence without having to unzip the double helix. So unzipping double helix is not required. So option number E is also wrong, right? So the final answer is option number A. And that's it for this question. Thanks for watching.